Mycobacterium tuberculosis is classified according to the Advisory Committee on Dangerous Pathogens, the ACDP, as a Hazard Group 3 organism. All work on Hazard Group 3 organisms needs to be performed in a Containment Level 3 laboratory. The Containment Level 3 laboratory is a self-contained laboratory unit maintained at an air pressure negative to the atmosphere, that is, at negative pressure, for optimum continuous inward airflow. The negative pressure is achieved using a dedicated air supply and air extract system. The air supply system will alarm if the negative pressure in the room is not maintained. The air extract system is provided by the Class 1 safety cabinets, through which airflow occurs via high efficiency particulate absorption (HEPA) filters. The negative pressure in the room ensures that the airflow is continually pulled towards the cabinets and the HEPA filters. All investigations on samples within a Containment Level 3 laboratory are performed whilst working in the Class 1 safety cabinets. The airflow through the entry of the cabinet is maintained at a rate of between 0.7 and 1 meters per second, and therefore the biomedical scientist is protected from any aerosols generated from the manipulation of samples. Additional requirements of a Containment Level 3 laboratory include limited access to authorised trained personnel only. The lab also needs to be completely sealable to permit fumigation in the case of a hazardous aerosol accident. The lab needs to have its own dedicated equipment, including personal protective equipment, non-opening windows, its own wash basin, and an observation window so occupants can be seen. All waste generating from a Containment Level 3 laboratory also needs to be sterilised by autoclaving prior to removal from the department for incineration. Before the biomedical scientists can enter the Containment Level 3 laboratory, they need to remove their day-to-day white laboratory coat and change into a dedicated green lab coat that fastens at the neck and has elasticated cuffs. This green coat is changed regularly and remains within the Containment Level 3 lab and can be easily removed in the event of a spillage. The most common sample sent for the investigation of Mycobacterium tuberculosis is the sputum sample. Mycobacterium tuberculosis is a slow-growing organism, and other contaminating normal bacterial flora in a sputum sample would occur if the sample was cultured directly. Sputa and other samples that contain contaminating bacterial flora are decontaminated with sodium hydroxide prior to culturing for mycobacteria. An equal volume of sodium hydroxide is added to the sputum sample, mixed on a vortex mixer to emulsify, and then left to decontaminate for 25 minutes. After this time, the sputum and sodium hydroxide mixture is then centrifuged at 3000 G for 15 minutes. This is 3,730 revolutions per minute. After centrifugation, the supernatant is decanted and the concentrated deposit is resuspended in phosphate buffer to neutralize the sodium hydroxide, and it is vortexed and re-centrifuged. The final buffered deposit is inoculated into a liquid culture media and a solid slope culture media. The liquid culture media is a commercial-based system for mycobacterial culture. It is known as the BACTEC Mycobacterial Growth Indicator Tube System, with the acronym MGIT or MIDGIT. The MIDGIT tube contains a modified Middlebrook culture broth and an antibiotic supplement. Growth of mycobacteria depletes the oxygen present in the medium. There is a layer containing a fluorophore at the bottom of each midget tube, and reduction in oxygen level activates this. Once each hour, the BACTEC midget reads every tube in the instrument and compares the readings with those taken previously. If the degree of fluorescence has changed significantly, the tube is flagged as positive. This usually occurs between 10 and 22 days. Those tubes which remain negative for 6 to 8 weeks are reported as negative. Lowenstein-Jensen slopes, LJ, are the main solid media used to culture mycobacteria. It contains inspissated eggs, malachite green and glycerol, and some slopes have pyruvate added to them. LJ slopes are incubated for 10 to 12 weeks at 37 degrees and need to be examined weekly by eye, with positives usually appearing much slower than liquid culture, between 23 and 30 days. The smear made from the decontaminated sputum deposit is stained by one of two methods: the auramine phenol fluorescent stain or the Zeil-Nielsen ZN stain. The cell walls of mycobacteria contain a high concentration of lipids, making them waxy and hydrophobic and impermeable to routine stains such as the Gram stain. They are also resistant to acid and alcohol and are described as being acid-fast bacilli, AFB, or acid-alcohol-fast bacilli, AAFB. The auramine phenol stain procedure involves staining the slide with auramine phenol for 10 minutes, washing with sterile water, and then adding 1% acid alcohol for 5 minutes, and washing again with sterile water. The slide is then counterstained with 0.1% potassium permanganate for 30 seconds. The auramine phenol penetrates the cell wall of the mycobacteria and is not removed when exposed to the acid alcohol. The acid and alcohol fast bacilli then fluoresce yellow-green when examined under a fluorescent microscope at a ×400 magnification. An acid-alcohol fast bacilli positive smear is a very significant result for clinical patient management. Positive patients require prompt anti-tuberculous treatment, infection control isolation if they are inpatients, and tracing and treatment of contacts needs immediate follow-up. An alternative stain is the Kinyon cold ZN stain, which uses Kinyon's carbofuchsin for 10 minutes, which has a high concentration of phenol in it, and it does not require heating as you would need to do for a regular ZN stain. After the carbofuchsin, 3% acid alcohol is added for 5 minutes, and then counterstained with malachite green for 30 seconds. Acid alcohol fast bacilli appear as red rods against a green background when viewed using oil immersion at a ×1000 magnification. Isolates of Mycobacterium tuberculosis on Lowenstein-Jensen slopes appear as irregular, dry, biscuit crumb colonies, which are buff beige in colour. When a midget liquid culture bottle flags positive, which usually occurs much more quickly than the solid culture growth, some fluid from the bottle is removed and it is stained using the ZN method to confirm the isolate as an acid-fast bacillus. After confirmation, some of the liquid culture is sent to the Mycobacteria Reference Unit, Health Protection Agency laboratory in London, where they will confirm the identification of the mycobacteria using nucleic acid amplification technology. They will also perform susceptibility testing of the isolate to a range of anti-tuberculous drugs, and they will type the isolate for strain epidemiology.